This presentation will throw light on the propagation of a nerve impulse along the axon of a neuron. John Carrier, Alan Hodgkin, and Andrew Huxley shared the 1963 Nobel Prize for discovering the mechanism of nerve impulse transmission. Neurons communicate with each other through an electrochemical process. Every time you think, learn, or communicate, a neuron in your brain sends a nerve impulse down its axon. The axon of a neuron makes multiple thousands of connections with many thousand other neurons. To understand the mechanism, let us first have a look at the plasma membrane of a neuron. Like any other cell, the neural plasma membrane also have the famous fluid mosaic model having the phospholipid bilayer. They have these green sodium-potassium pumps which help to maintain the potential difference. In addition to this, they also have voltage-gated yellow sodium and blue potassium channels. Here we connect a voltmeter across the membrane to measure the potential difference fluctuations with respect to time. Initially, when the neuron is not stimulated, its membrane is polarized, which means the outside is more positive and the inside, that is the cytoplasm, is less positive. The presence of negatively charged proteins and nucleic acid in the cytoplasm is one reason for this difference in charge distribution. The outside contains excess sodium ions and the inside contains excess potassium ions. The membrane potential is minus 70 millivolts. When a stimulus is received by the dendrites, sodium ion channels open and sodium ions rush into the cell. A stimulus can be pressure, heat, light, or chemical information coming from other cells. The neuron at this stage starts depolarizing. If the stimulus goes above the threshold level, more ion channels open and more sodium ions flood into the cytoplasm, which in turn activates other sodium channels to open, and this goes on and on. This leads to complete depolarization and the membrane potential increases. Once a large amount of sodium ions enter the cell, the sodium channels close and the potassium channels open, with the potassium ions moving outside the cell. This is called repolarization, and the membrane potential starts to drop. At the end of this step, when potassium channels close, the electrical balance is almost restored, but we see that the membrane potential is slightly less than minus 70 millivolts. This is because the potassium ion concentration on the outside is actually slightly more than that of sodium ion concentration in the cytoplasm, a state called hyperpolarized state. Here is a comparison between the initial polarized and the present repolarized state. To convert it from this state to this state, during the refractory period, the sodium-potassium pumps work rigorously. Once the impulse has traveled through the neuron, the membrane returns to resting potential minus 70 millivolts. During the refractory period, this is the initial shape of our green sodium-potassium pump. Then this pump takes up 3 sodium ions from the cytoplasm. ATP molecule bind to this pump because this is an active process which requires energy. The energy released by the ATP molecule is utilized to change the shape of the pump. The 3 sodium ions are released on the outside of the cell. If the phosphate molecule leaves, the pump will regain its original shape. But before that happens, before the phosphate leaves, 2 potassium ions from the outside bind to the pump. It's after this that the phosphate group leaves and the pump regains its original shape, and the 2 potassium ions are delivered into the cytoplasm. These pumps work rigorously during the refractory period, and there are so many of them on the membrane which keep pumping sodium and potassium ions in 3:2 ratio. After the refractory period, the neuron is back to polarized state and stays in that state until another stimulus comes which can cross the threshold to generate action potential. And all this happens in just few milliseconds. Thank you for watching the presentation.